0.1? That's pretty slow.
Well, how fast do you want to go?
I don't know, 0.3, 4, 0.4? How far was it? 100 meters?
I called in a step for 50 meters. We're going to eventually go 114.
That's not very far. Okay.
You're happy with the speed?
At 0.1?
0.1.
No, you should go quicker than that.
What?
You should go quicker.
Okay, how fast?
0.3.
Bridge nav.
We're
Can
not
you
gonna stop or nothing.
go
Right.
0.3 knots? Thank you.
It really does look like confetti.
But if that confetti hit you, it would stick to you.
It doesn't sound pleasant.
Like the mud that splashed on us.
Oh, yeah.
Oh,
Yeah, I'm still covered with mud.
Yeah. Don't look at me.
Huh?
That's not me.
You?
That's an
yeah,
individual.
how has the lab
That's
processing
different.
been
They're talking about something
going?
different.
Talk to us a little bit about what's happened in the wet lab.
Oh,
Yeah.
the processing has been going, great. We've been sectioning the cores and we've been picking. Joan has been busy. That's why she's not here. She's down picking 4Ms and from the sediments from the last dive.
What is the tool you use for that?
Well, she uses a dissecting microscope and little tiny petri dishes, and she sieves the sediment and to separate out a lot of the fine silt to get rid of it. And then in seawater, she picks using a pulled pasture pipette, a very thin glass pipette with a little bulb at the end. But she uses a sort of a mouth pipette system, where it's very finely controlled. And I'm sure you can find some pictures of it on the website from today. And we were just laughing about mud on us because for one thing, I wanted to section a core and we didn't have fantastic luck with that core. We were transferring it from one puck to another outside at the wash basin, and it kind of blew up all over us. So
When you're picking, are you picking 4Ms out, or are you picking dirt out or sediment
no,
out to leave 4Ms?
We're picking the 4Ms out.
Does anyone know what these like cylindrical?
So this to me looks like modern art. The bottom here with these mats and the different colors. It's really quite beautiful once your eye adjusts to the subtle color differences.
Could be kind of like abstract wallpaper.
Yeah, like what's see this? Huh.
Is that more kelp? Probably, huh?
I don't know.
I'm gonna stop and look.
Yeah.
Video zoom.
Bob, is that a zoom?
Yeah, well, it's a zoom.
It's a Kylie's
I'm flying.
Oh, sorry, I didn't notice the change, so that's why I was like,
Is it kelp?
Yeah, probably old kelp.
Old kelp? Kind of looks that way.
Dinner bell ringing. People wanted whale fall, we got kelp fall.
Yeah.
Vegetarian version.
But it was kind of laid out in the shape of a whale.
Yeah,
A little bit, yeah.
So
the ocean's trying.
Video come wide.
So Ginny, I have a question for you, something I've been wondering this whole time, and you've probably answered it a million times before, but we hear a lot about dead zones, at sea, and dead zones are usually thought of as kind of a negative thing, you know, tons of fertilizer washing into the Gulf of Mexico. But this is kind of, is this a dead zone? And obviously there's life here, but is this, I guess my bigger question is just like, is this bad for the ocean? Is this good for the ocean? Is it not that simple?
I don't think it's that simple because dead zones are bad for certain organisms. Well, first off, they're not dead. Okay, they're dead for things that people care about, fish and macrofauna. You know, there's a lot of animals that cannot tolerate, most cannot tolerate such conditions. So if bodies of water become consistently low oxygen, it drives away the larger animals. And because fish can't live there, if it goes anoxic, people call them dead zones because they're dead to fish and other critters. But there's plenty of microbiology that goes on there. It's just not the kind necessarily that benefits the food web as we know it. So you might start to favor certain types of microorganisms that produce a lot of sulfide, for instance. Then that gives the water that sort of rotten egg smell of because they're carrying out that the way they make a living is that they are, you know, respiring sulfate in the water and producing hydrogen sulfide. And that is another sort of attribute associated with dead zones, quote unquote, is the sort of smelly, fishless, animal-less zones. And of course, those aren't good in the grand scheme of things to
Okay, ship is at its position.
Expanding zones of where fish can't live. I mean, there's a lot of naturally occurring little areas like that, but they're not meant to be in the midwaters of our big oceans, for instance.
Well, no, we're not there. I want to talk with science for a minute. So I just want to make sure we're all situated because I understand this is really light sensitive. Yes. We are currently 73 meters from those cores, so we're outside the 50-meter buffer. It took us about 6 minutes to travel that distance. So right now it's 1:47. If we continue on, we'll be at the cores before 7:05.
Okay, so let's slow down just a little bit and just maybe rest here. And what we'll do is when we get, as we approach the cores, I say we turn off whatever lights he doesn't need, you know?
We could turn some off
and,
now.
you know, well, you know, if people would like to look at what's going on, you know, and while we're hanging out.
Probably get rid of some, though.
Yeah.
Still.
But when he's actually picking up the cores, you know, so this is great, you know, but when he's picking up the cores, he may need more light.
Yeah.
So.
Okay.
He should use whatever light he needs, but not more than that.
Ooh, this is sort of nice mood lighting.
Yes.
Okay.
This looks like Mystery Science Theater.
What time did you want to start picking up the cores?
We want to start picking up the cores, I think, at 7 o'clock.
At 7 o'clock? Okay.
Yep.
Bob, how long will it take you to get situated when we're at the site?
Oh, man, you guys are just making me throw out all these numbers.
Yes, we are. You have to work today.
Well, I don't know. I mean, it shouldn't take very long to get set up. As long as we find the thing quick. I don't know. I guess we're going to have good nav. Yeah. I mean, do we know the bearing that they were on when they put them down?
Yeah, I hope so.
Throw some numbers back at you.
Yeah, right back at you.
I'm sure that the computer has that information somewhere. We have to look for our footprint and then just land there.
Yeah. In the dark. Yeah, we do want to land like where they were.
So when we do
the bearing they were on when they were put them down.
When we do the coring work, do Argus lights go off also?
Yeah.
Okay.
I think, well, they were using some Argus light, I guess. We're just going to have to see.
Do you want to see what it looks like now?
Like he's not going to be able to place them in the quiver with this light arrangement, I don't think.
No.
Roger.
No, I'm not.
Well, you have your starboard bio.
He's
bump.
good,
They're in the front.
but-
Yeah.
Well,
Just
and-
shadow puppet
I mean,
version, yeah.
Tammy can turn up the iris. And,
Yes, I can
Yeah.
also play with the gain and everything
yeah,
here.
so see what it looks like. I mean, this isn't as
Oh, look at that.
Whoa.
Yeah, if you try, you have too many lights on.
so how many? Well, okay.
Give it a shot. I think you'll see that we were able to crank it up to 28, Tammy.
I'm not like right under Argus, so
Now,
Well, Argus lights are off,
Maybe one more. What was that 30?
oh,
That's 42.
they're off.
Yeah.
so I can give you Argus
Oh yeah,
light.
Argus lights are off because we get a better view of where our position is with Argus lights off. But, so this is just the side lights and the aft light. I don't really need the aft light, I guess.
Tammy, can you crank up
This
the
is-
pedestal?
I can't
a little
work
bit?
This looks dicey.
Yeah, you think? I can do the porch light. Turn off the other lights.
Oh, that's nice.
That looks good.
Wow. Just sitting on the seafloor with your...
That reminds me of like a summer evening on
Right.
the porch.
I mean, I'm not really liking that a whole hell of a lot.
You just need your bug zapper.
is the 7 o'clock based on how long the cores have been incubating, or is that working back from an on-deck time that we
That's
want?
working back from an on-deck time.
Gotcha.
But is the on-deck time critical, or is it just because we're trying to maintain the watch thing?
I think it depends on who you ask. I bet if you
Yes,
ask
I
Mark, it's
think-
critical.
Is it trying to not be earlier than that, or trying to not be later than that?
That was the message from the boss.
Mm,
Yeah.
So
well,
we're on
it
board.
is like our
We're on
I
board
can
with
I
that.
can go confirm that whether that's flexible or what the
well, if
whether
we-
there's a hard stop at 8, because it looks like this is a good time for me to.
Find out, yeah.
Yeah, I mean, if we have to meet a boat somewhere or something,
Yeah.
I understand. But if it's just like we're just trying to stay on the watch schedule and it's the last dive, we don't.
I think it's their lights, isn't it?
Well,
Not
we
to
can
expose
be
it to the
after
sunlight?
8, I'm saying.
Yeah,
We're going to water.
no idea.
So our goal right now is to creep up on these samples
Mm-hmm.
as darkly as possible.
Yeah.
And so do we come to the surface with
And-
all of our lights off?
And then there's going to be some more cores. And then we're going to get those cores.
Okay. This is
Then we're gonna
one,
run away.
One,
And we're going to try and do that all.
one little incandescent
Very dark.
bulb on, so it's it's about as low light as we're gonna get.
Kylie, are we
Yeah.
out of auto XY?
Yeah.
We're out of it?
Well, yes. Yeah, I was fine at normal. Yeah.
Thank you.
Bob, you're popular tonight. We got more questions for you.
Oh.
Hang on one second. Bob,
Uh.
is there a way you can use like Argus lights if Argus is over, and then I can bump up the iris and gain for you?
Let's see. Let's test it now.
Well,
Did that
get-
with
get
that
under-
All right.
under
abyss, and
All
it
right.
worked.
Give me a moment, please.
I mean, Argus's lights are pretty bright.
Yeah,
so
they are.
This is just one incandescent porch light, so
Wow.
this is pretty low light. It is closer to the bottom, but
I think what Tammy was suggesting was Argus lights on, all Herc lights off.
yeah, I'm saying the Argus lights are pretty darn bright, right? Those are
Yeah.
seriously bright lights.
Oh, you think they're too bright for the
Yes.
samples?
Well, I think this is just a little. This is the porch light. It's just one little incandescent bulb,
And this is with the iris all the way up on that.
and this lights up the things that I care about, which is the samples. I mean, I got to get them in there. I can't.
What if is Herc butt light on?
No.
Can you turn that on and turn porch off just for a second? Just
I think
curiosity.
it's too
Oh my gosh,
noisy.
I forgot what I'm doing. But light, that's that.
F1.7, Tammy. Gain.
Where's the butt
40.
light? Mid down, starboard port aft. Got it.
Nothing.
Is that
Oh,
Bud
that-
Light?
Yeah.
Oh, yeah.
Yeah. Okay. Oh, and that was with no Argus lights, eh?
Right.
Yeah.
I'm just trying to position.
You want to see what it looks like with Argus lights only? Is that the deal?
Yeah,
Sure.
I was trying to move to get actually under
You
you.
want all the Argus lights?
Sure.
All right. Well, that's
And then the porch off.
That'll work.
Is that good for you?
no.
And then turn your pedestal up, your blacks. It's a knob to the
What
left
if
of the
I
iris.
turn my heading to
Yeah, it's up all the way.
like face Argus, so the Argus is like not looking at not lighting the butt of Hercules, and it's like
I just think that we're gonna end up with a shadow because the overhang of the brow. You know?
Yeah, Roger.
In the places that I need to be able to see what I'm doing.
Yep. Okay.
Yeah, I think you're going to have a hard time putting a quiver this way.
Yeah.
I think porch is your only option.
Yeah. I think porch would be the best because it's one incandescent light. You know.
Does that work for
Okay.
you, Jenny?
Yep.
Great.
All right, so you guys have your lighting situation decided upon?
Porch. Let's turn that on now.
I'm going to call in a ship move, and we're going to head to the place where we can pick up the course.
Mm-hmm.
Okay, we can kill the Argus.
Go for it.
Okay. Argus lights are off. Porch light is on. We're going places. Big eye. I'm up on
This
my
is nice.
delta
It's
because
a
I'm like
nice
under you.
Light.
FYI. And ship move whenever you're ready.
Okay, 64 at 207.
I do need...
So Jenny, what is the plan when they get up to the surface? Are they going to like go inside a black box, or are they going to be immediately fixed? Or be fixed and go in a black box?
They'll get taken off the porch here and go immediately into a black bag.
Okay.
And then one at a time, we take them out and we fix them.
Bridge nav.
And what does fix mean for everyone at home?
Preserve, stop, kill.
Get like a snapshot of exactly where they're at that point.
Yeah.
Freeze. Freeze tag.
Yeah.
That's good.
171.
Kind of hard to see them
Yeah, and
down on
you
the
can
seafloor, you know.
keep the speed of
What you doing?
She said 171.
0.3 knots.
Are you doing with Argus?
I'm going to do both. I like when
Okay.
they face the same way. It helps me
Thank you.
keep it in the middle of the screen.
Okay.
I want to do both. Okay.
Do you feel like you need a little bit more light out front in order to
see where it is?
see where they are, so we
Not
don't
drive into
just
it.
mow them
I
over?
still didn't get an answer as to what the bearing was when we took they put them down.
Oh, I haven't. It's not. It wasn't recorded.
Nobody wrote that down?
Do you
Okay.
have that?
Should I?
I don't like that.
Well, it would, I mean, if we want to set up quick, then it would be best if I came in on the right bearing.
I turned the mid on so we can come up on it, and we can turn it off as soon as we see them. And just use porch as the only other light that's on.
Yeah, I don't know.
I just don't want to drive into them.
Oh, you're
Yeah,
choosing
I would say
yourself.
keep it like this until you see them, and then you know where they are.
Exactly. Or yeah, as we're getting closer,
Yeah.
we'll be able to. Yeah, exactly. We have a mark for them, right? Yes, of course. Okay. So how
I
we're
can
still
go ask Nicole.
we're still like what 70, 65 meters away, something like that.
We're 51 meters away.
Okay, Roger.
All right, so we're stealthing up on these cores. And then you'll see some more lights go. We'll land, see some more lights go out, pick up the cores. Just a couple more objectives left. If you have questions for the team, send them in now. Nautilus Live, we'd love to answer them for you. Bob, some questions about, could you talk a little bit more about, we're on the ground fault question again, about how electrical current makes its way through seawater instead of through the chassis is the question. So could you talk a little bit about that?
You got to have a complete circuit, right? So one of the legs of the path back is the chassis of the instrument or whatever.
Mm-hmm. What draws the most electrical power, about lights or hydraulic pump or?
The hydraulic pump is our biggest power draw. So we have a 20 horsepower hydraulic pump, and it runs on the three-phase AC power down the main cable, and that's 2,600 volts. And so we're currently running about, the vehicle's like 36 amps per phase. So that it's by far the biggest energy draw. All our lights are LEDs, or almost all our lights. The porch light's the only like incandescent bulb. Mm-hmm. It's a halogen light. Or wait, this one is LED.
I don't know.
There's a halogen light on Argus, but not on Herc. Oops, too low.
You're too far down, man.
Coming up.
Okay, Bob, I'm gonna change us a little bit.
Yeah, how far we got?
Bridge nav. Can you change our current move to 4-3 meters at 235?
That's.
Yes, please.
That's the bearing.
235.
235.
Mm-hmm.
And Bob, I don't have the I don't have that heading for you.
Yeah, okay.
It wasn't recorded.
How far away
oops, now? Oops, oops, oops.
$45 meters.
235. Okay, FYI, 0.7 tether wraps, just.
Where's it at? Like, where's it at on here? I can't see that, so it's like not very handy.
I can see what you mean, Denny, about like the abstract shapes. I'm kind of like imagining, you know, when you're on a plane and there's just like textures and patterns and.
Yep.
All right.
When you're looking like, down. Yeah.
Yeah, I can kind of envision that I'm.
All right, give me this back.
15,000 feet, 20,000 feet in the air, looking at it.
In the right direction.
Yeah.
FYI, oh, okay, the tether wraps are better now, but okay, good. You're centered up.
Like scale is such a funny part of this. I always, you know, when I fly home from an expedition, and if we've been diving somewhere, you know, somewhere on a seamount or something like that, you know, and then you fly home and you're flying over big features. I live in the Pacific Northwest where you have big mountains, and I'm like,
you're looking at a big mountain. You're like, wait a minute.
I was just there,
yeah,
but it was hidden.
Yeah, you were looking,
but it was underwater.
Mm-hmm.
Sorry.
Yeah, hiking in the Rockies with a flashlight, you know.
Good.
Cool.
Do you have an idea of how quickly these, what the sedimentation rate is here? That one.
Boy, I'd bet it's millimeters per year. It's pretty high, you know, because look at all the stuff in the water column. I don't know for sure, but if I had to guess, I would say it's really high like that.
Do you think our Herc footprints will be colonized by a bacterial colony?
Oh yeah.
Taking advantage of that space, or
yes.
covered by sedimentation? Which will happen first?
Probably the mat.
Okay.
Mat growth. You know, if you come back, I don't think you'll see Herc's footprints in, you know, a couple months.
Wow. 12 meters away.
Where?
You're about 12 meters away from it.
Oh, I am.
There's some piece of wood, like a paint stick.
So the seafloor here is basically collecting a lot of the stuff that's flying up in the air right now. All that marine snow is just coming down to rest here.
Yep.
Blizzard.
Well, some of the stuff that you're seeing is swimming, right?
Yes. On your left.
Yeah,
as well.
it's not ready to sink to the bottom.
It's not this one.
No, not yet.
It's this one over
That
here.
looks like an old footprint. Is that right, Bob?
I think so.
Yeah, I mean, looks
And
like
maybe another one to your left?
Yeah, there's two spots here. Which one is it?
Well, we've got to see the tops of the cores.
So there's a lot of critters that have moved in over here. Like maybe that means it's
What are those?
fresh.
Are those iris zones?
I
can't
think
tell
I saw the cores to the left.
To the left?
Watch in the left corner.
Yeah.
You see those
Up
right
there.
there?
Yeah.
Oh,
Little
yeah.
reflections.
Okay.
Oh,
Yeah.
yes.
You
I'm
want
going to turn
the
off the mid-light.
off.
Oh
Whoa.
my God.
Wait a minute. No. No, it won't be a good day if we just sit on them.
I see them.
You want to back on?
No, it's not on it. That's on Tammy with the iris, right? Iris, yeah.
Correct. Iris and gain.
We're just on the porch light.
And the orientation of these is kind of like an L. I don't know if you were shown an image of how they're kind of
They're
laid
also,
out.
they're in your bubble cam now.
Oh, over.
Well, here we're coming right up on two of them.
So one thing that
The rest are
right
in the
next
line.
to each other?
There's 1, 2, and then there's 3 in a line behind them. You see them going kind of like
yes, this is
northeast.
the perfect orientation. Okay, so I can tell which sample's which. Because yeah, that I'll need to know which sample is which from our last notes, and we wrote a little diagram. So we want to make sure that we're staying with that so we can log the proper sample numbers and stuff. That's mostly on me, but with cooperation of the pilot.
Don't sit on them.
Yeah, depth perception is not great here.
you're right on. You've got like a foot in between them, so I think that's probably safe.
So there's two here. Is that
There's 2, and then do you see 3 more in the
I
foreground?
see 3 more in the distance.
Yep.
Yeah, okay.
Okay. Wow.
There was 5 total from the last dive.
That's my understanding.
Yeah, there's
yes.
Okay.
Now I see the T's. My eyes are adjusting. I was like, oh my God, there's no handle.
Yeah. Yeah,
Somebody
so these
stole
two
the handle.
Stole the handle.
So
If you were to do these again, would reflective tape, like on the handle or something like that, would that affect it at all, or what do you think? Because that would probably be really handy for
I think so.
and the ROV.
So you've got 2 positions up front, Bob, and you've got 2 on the right, and 2 inside, or 1 inside.
Yeah, I don't know about this lighting. I think that's... You got a strategy to give me better depth perception?
Hey, Tammy, can you crank your pedestal down, give him a little more contrast?
That's better. Yeah.
And just like that, the T handles appear.
Can we zoom in on that? And Kylie, can you be my tilter? Okay. Ready to zoom out? I think I'll go in this one on the right there.
You're completely racked out for the camera, FYI, I think.
Oh,
yeah. You want it in?
Yeah, come in a little bit.
Okay, coming in.
That's good.
That's good, okay.
Okay, ready?
Yep.
Go. Hey, can we zoom in a little?
Great one.
Okay, tilt up.
Very nice. Great. Looks like we got one, so we can
now let's work on this next
go home without shame.
I can't see it.
What was our acronym? Extreme Pulse Indicator Device, the EPID. Yeah.
How's that?
Yeah, this would not be time for the a blood pressure test.
Okay, so I'm going to go for the other one on the right. So you ready?
Yes.
Yeah.
Great.
Ooh, a bunch of pyrosomes caught on for a ride too.
Yeah. Got that. Maybe one will come up with us. Doubt it.
A couple came up yesterday.
Really?
I
would love to see one. Do they stay kind of chubby or do they flatten out when they?
No, they stayed chubby. Kelsey compared it to a, Kylie compared it to a gummy worm, like a texture of a gummy worm. And I thought that was a good comparison.
Yeah.
Okay, so now we need to sort of float up and forward. Is that the plan?
Yeah.
That was great, Bob.
You got a lot of mud in on your vehicle.
Yeah. Wah, wah, wah.
Can't really see the orientation of these other ones.
I think the heading is
bubble cam.
twist, turn to port.
Can you see them?
I can see them
Sorta.
I see one.
And in the bubble cam.
See this view down here?
Oh, yeah.
That's the one looking straight down.
Oh, I see them now. So does that fish.
Oops.
Ooh.
right up on one of them.
While we let this settle out, someone wants to know what does the substrate feel like in your hand? What is the texture? Looks super gooey. Is that what it feels like?
No, it feels wonderful.
Okay,
it's not gooey at
all.
all. It's soft and silky.
Oops. Okay.
So if you have ever had a mud facial or mud, it would be like the most expensive mud facial because it's because that would be accurate. Yeah, exactly. because you brought the Nautilus out to get it. Yeah,
sorry,
but
sorry. Can we get down for a second?
Of course.
Can you zoom in more? Trying to see the T handle there. Yeah. Okay, so I'm going to shoot for the center gap and come to the back core, back on the port side.
You want the tool tray out, like when we get to that?
I don't think I need to.
Okay, I'll just drag.
Okay, you guys ready?
Yep.
Yep.
There you go. Okay, zoom out some. Yeah, we don't want to go tool tray out anyway because there's cores right there, front.
Okay.
Oh wait, I want to go to this one. I think.
Oops, sorry. Opposites. Opposites.
Nice.
Huh? Now what?
Yeah, can you see any of the others?
Yeah, very nice. You made it look easy.
There's one directly in front of that quiver in the middle.
Oh, that's handy.
There's
That's, well, you might need to back yourself up a bit.
But I want the purple teddy bear.
Oh.
You can see the T handle, yeah.
Can you zoom in on that? See
sure
if you can see it.
You can see both of them.
Did you rack all the way out?
Oh, look
I'm
at
pretty
that.
much all the way out, yeah.
Yeah, you
I
can see the
give it a
T
little
handle.
more. That's all I got for it. Out.
Whoa.
Look at the shadow of it.
Yeah, that was scary.
You want a little more zoom, Bob?
I think it's all right.
Okay.
Oh.
Okay, maybe we'll put this one right up front there.
You're on the lip of it.
Beautiful. Oh my gosh.
Okay.
Ready for zoom?
Yep, zoom.
It's so good.
Got more zoom.
It's worse now.
Oh my God, this is like blind.
Can you tell
He's
if I'm
got
in
Yeah,
there?
yeah,
Yep.
We've got it.
yeah.
Holy
Okay,
moly.
this one, I think we'll go in the slot and in the back hole.
Okay.
Okay, ready? There we go.
Look at those nice layers. Look at those layers.
They look great.
Like a dirt cake.
Ew! That's great.
Yeah.
Oh, yeah.
Wow, that's amazing.
That was the last one.
Yeah, that's it. We can turn the lights down.
Wow.
That was awesome.
So
Crushed it.
For lights, we just need to be set up for the Niskins.
Nope, you're now collecting Niskin bottle water for DJ at selected depths, which I don't have. Okay, so we've got it covered.
So do you want to put your arm like on the Niskin and then freeze it there or something?
Joan says yay.
Okay. Tell Joan it took
Can you hear the cheering?
It took a couple of years off my life.
Yeah. You can cut the tension in here with a knife.
Oh my God.
There on the bottom.
Equal spread of like lean forward people and lean back people and just like.
Oh my God.
I gotta have some light.
That's great.
Yeah.
Yeah. Nice job, Bob.
Yay, Bob.
Hey Taylor, what is this Niskin sample going to be that we're taking on the bottom?
God.
Do you want to?
No, you need to go have like a mud facial in all of that.
Yeah.
Do you want to get oriented
in?
for a recovery, or do
I
you
just
want to set
know
up
the
your
sample.
arm for the Niskin first?
We'll get in recovery position.
Okay.
Yeah.
So I need lights on Argus. I need at least one. And I need to figure out where you are in relation to me.
I thought afterwards I do this workout video where this guy is always saying no turtling, you know, like when you're tense with your shoulders.
Yeah.
I was just like, wow.
Like the full turtle.
Full turtle.
Yeah. Yeah.
Did we take that Niskin?
No.
Are we supposed to take one at the bottom?
Yeah, we're supposed to take one at the bottom.
We're still on the bottom, I think.
Yeah, DJ was just coming in.
Sorry, I was on mute. I was talking to Nav and not talking on SPL, so you didn't hear me.
Here he is.
But yeah.
Oh yeah, we're actually doing really well. We're exactly on time.
Imagine that.
I don't know.
Yep.
We gotta take Niskins.
You want
We're
one
doing
at the
Niskins
bottom.
on the ascent though.
You want one at the bottom?
Yeah. There's one at the bottom, right, DJ?
Right.
Okay,
at the
yep.
bottom?
So, take one now, and
Yeah.
then we'll float up.
Let's get a light.
Yep,
on.
Roger.
Oh, come on. You know where it is.
Okay, camera back.
For viewers who are hoping we'll go back to the Gulf of Mexico, we have been there. We've dove there a lot. If you want to check out the highlights from those dives and see some of our discoveries from there, you can actually search the gallery page by the... DJ, can you mute?
Somebody's microphone.
Can you mute while you're moving her mic around? Thanks. Got it. Oh. Yeah.
How many Niskins altogether, DJ?
I was told that there are only 5 that we can get on this dive because one of them's malfunctioning. Is that correct?
Fixed it.
Yeah,
Oh, you fixed it.
Yeah,
Okay,
it's
so
fine.
then 6.
Somebody cross-threaded
The air vent.
Oh, okay.
Which one of these are you pulling right now? Is that the last one, 6?
last one.
Yeah.
You ready?
Yep.
You see it?
Yep. Good.
Okay.
Great.
I
Do
will
you-
get set up for this next one.
Do you have the full depth plan, or
You'll have to tell us.
do you want me to tell you as I go or all at once?
Can you give me a heads up now? I think the
Sure.
the most shallow one is 400, right?
Yes, the most
Okay.
shallow is 400.
So there are 5 more between now and 400. Okay, and all right, go ahead and tell me what depths they are.
550.
Yep.
525.
Yep.
500.
Yep.
450.
Yep.
And 400.
Roger.
Okay.
Okay. So we're at 565, so we'll come up slow and pop this one in 50 meters.
Light out.
Light out. Okay, I can turn off the one on Argus.
Eh?
What? Oh. Okay.
Okay, I'm driving out and then I'm coming up.
Okay, I will turn off my auto heading now. Is that okay?
Just a second.
Okay.
Okay, you can do that?
Yep. Okay, auto heads off.
I didn't put it on. It made it really bouncy. Yeah. That's no good. No good, man. Okay, I think we should turn on the Argus butt light, though.
Okay, Argus butt light. Oh, yes, tether. Yeah, yeah, roger.
There you go.
Bridge nav. Just an update that we are starting our ascent.
Yeah. Thanks.
We're coming up.
We're coming up on 550, which is the
Roger.
next one. Yeah.
Stand by.
That's
Okay.
the next one is
Right,
550?
right now, yep.
Okay, light.
Light, mid, porch.
Light.
We're at 550 meters.
550, yep.
Got an eye on it. Good, it popped. We're good.
Bingo.
Popped.
Yep.
Okay.
Next depth is 525.
Okay.
And which handle did you just put the green one? I can't remember. Yeah, right? Green.
Lights. Oh, I don't think.
Ready for light?
Lights out.
Lights out.
Can you hear me?
Okay, coming up.
Yes.
Yeah,
Oh, okay. I got you now.
Yeah.
It takes like being up that high for me to hear you on your mic. Yeah.
That was the green handle you pulled, so position number 5.
Yeah.
That's correct.
Just double checking. 095.
What would you be most surprised to see when we turn the light on right out in front?
Boy, there's all sorts of things that come to mind. A large octopus stealing my cores.
Jacques Cousteau.
Or just the handles off of
Yeah,
them.
yeah,
Unscrewing just the
exactly.
handles.
That's it, right.
That's where all the gaskets went.
Yeah. Yeah.
525, right?
That's correct. 10 more meters.
Yeah.
What's the name of that guy on Argus? Billy Argus?
Bobby Argus.
Bobby Argus.
Bobby Argus.
Yeah.
I'd be surprised to see Bobby Argus.
Yeah.
Okay, 5 meters.
All right, yep.
You maybe want to fly forward a little bit more?
What would I be most surprised to see?
Light again?
Yep, roger, light.
Some creature
Port
stowing
on.
a ride. Oh,
Oh,
they're still there.
there.
525.
Eggs are still in the basket.
Watch it.
Whoa.
I think you already popped it. I think you just popped it. It's closed.
Yeah.
Is it?
Yeah.
Okay.
Okay. 525's a go.
Okay, porch side off. Oops, sorry. I'm sorry. I'm sorry. I got too ahead of myself.
Sample number
Okay,
096.
ready.
Okay, where's this one going to be?
It's going to be at 500.
Okay. Kill the light.
Lights off. And you'll pull forward a little.
What's that?
We're not like,
We're hoping to
we
get
have like a little bit of a catenary in our tether.
Push the stick.
Could you?
And can you push that stick forward a little bit? Don't push it from there. Push it from the bottom. Yeah, just push it forward.
Do it. Push it.
Oops.
It's not going.
And I was using the wrong stick. Like pointed at the sky now. Oh, keep going.
Do more.
You want to turn the light on for a second?
Yeah, porch. Porch is on.
Oh my God, they're missing. Oh, there they are.
Okay, light off.
Lights off.
Scooby-Doo and the mystery of the stolen cores.
Yeah. Yeah, well, you missed it, but when we first arrived, I couldn't see the T-handles. And I was like,
I'm no,
why are you going to pick them up?
500, yeah.
Somebody stole them.
Somebody stole them.
So what happens next when you get these on deck? Talk us through what the next few hours will look like for the team.
When we get them on deck, we're going to race over there as quick as we can and get them into the dark bag. And then one by one, we'll disassemble them and take off the first couple of centimeters and preserve them.
You got 2 meters, 1 meter to go.
Take off the first couple centimeters because that's the part you care about, or the part you don't?
We're gonna collect some of the water that's in the headspace above the core. Good. So that's right there. We'll collect some of that water into what's called an excitainer, which is gas tight.
Sample 097.
And we'll put that in the refrigerator, and then we'll take the sediments and we'll transfer them to a bottle, and we'll fix them with zinc chloride.
Okay.
And that's how we process these cores.
Zinc chloride.
Yes.
We use zinc acetate for everything. Wonder what the different chemistry.
Maybe it's zinc acetate. I'm not sure. But
I think I saw zinc chloride on the...
I think it's zinc chloride, yeah. Yeah, I don't know. So Dan Rogers is doing all the isotope analysis.
Oh, it's isotope analysis. Okay. Is it nitrogen isotope or
carbon?
Oh, that's right.
He said turn an orbit off when we want. I got that in a second. When you got that, where the next one's at
450.
450. We have 30 meters. So I will turn off an orbit now. Science. Orbit is off.
That yellow thing is not cooperative.
It's not cooperating, yeah.
Come on, baby.
We use
talk to Bob.
I want to be at 450.
450, yeah, that's correct. We're at 477.
Okay, there you go. Nope. Oh, you rotten thing.
Apparently,
there. Ooh.
He's got to get fixed. Really obnoxious.
Oh, man.
It's a hard game of the claw game.
Oh, yeah.
Why don't you try closing with it like that? Oh, never mind. Okay, 10 meters to go. There you go.
Wait. Can you start over? Wait.
9 meters.
Annie.
Yeah, sorry.
I don't think he heard any of that. Oh. I just turned on his audio so he can hear
54.
You.
Oh, God.
53.
Okay. Okay.
52. Okay, pull it.
50?
Now. Good, you're good.
Whew. That was hard one.
Next one is going to be
I heard
400.
it, but he didn't hear you. Yeah, I'm listening to you, but he wasn't listening to you. Yeah.
Okay, so I'll go ahead and turn off the porch light.
Yep.
Okay, porch light's off.
So wants to know if these samples are related to the clam samples. What is the connection between those?
Oh, the clam samples. You mean the samples of, the same or a similar isotope was used.
Okay.
Okay, so I'm not familiar with all the details of the clam experiment, but I do know that they provided an isotope and let the clams sort of incubate in the presence of that isotope.
They're also doing transcriptomics with the clams, which is to tell what sort of genes are active in the clams.
I feel like the big umbrella of this expedition is like, how are animals making their living? You know, how are they doing what they're doing? And we're asking questions about how are they doing what they're doing with little bits of oxygen? How are they doing what they're doing with no oxygen? How are they doing they're doing with like...
What's up with these chloroplasts?
Yeah, you like seem to have stolen some organelles from some other animals. Like, what are you doing with those?
When people ask what I study, I just say life at the extremes, which I think does summarize what I do, and it sounds really cool, so.
Good elevator pitch. Tell me more.
Under a microscope, what does a foram look like?
Boy.
What's the shape? You know,
what's the shape?
Yeah, you know diatoms are like a barrel.
Yeah.
What's a foram shape?
Well, there's so many shapes. Some look like a hot dog. Some look like an ice cream cone. Some look like a feather. Some look like a nautilus shell.
Wow.
Boy, there's so many shapes. Yeah.
Is that dependent on on the different type of species of
Yes.
Okay.
Yeah, they all have different shaped tests.
Oh.
And they're very beautiful, much like diatoms are beautiful with their different, many different silicate shells. So there's a lot of beauty in the protist world for sure. I bought a few old Ernst Haeckel prints.
Yeah,
and he did like these beautiful drawings.
I know of that.
They're really gorgeous art.
So interesting.
Last sample's at 400 meters.
Roger, 7 meters to go. Light going on. Lights on. 6. 5. 4. 3. 2. 1. Pop it. Good. Closed.
And that's all she wrote.
Stow the arm.
Taylor, and that was sample 099?
Correct.
Thank you. What rate are we ascending?
Give it a second. Give it 1 second. Time out. Porch light off.
Sorry.
Gosh.
No stress.
You're moving.
Right now it's 15 meters a minute.
Okay.
But that's not it.
That's
that was awesome. Everybody, thank you so much.
Yeah, thank you.
Yeah.
That was a good one.
Yeah, it really was a great way to end.
Yeah.
On a real high note.
Good.
Absolutely.
Okay. Just
what a great team.
Thank you, thank you. You know, Bob.
All right, now can you tell me?
I
gotta let the bridge in the deck now.
We gotta settle in. We just started.
Okay.
Takes a minute to 15.
Thank you.
Yeah, thanks for the question. Keep them coming as we ascend. It's a great time to get your questions in. A foram is a protist. It is not a plant, not a plant. We're a fish. They are microscopic little protists, sort of similar to a bacteria, but a little bit different. It's an ancient type of life. And